very good morning to all of you. I thank AIOS for giving this opportunity. And I'll be talking about the next frontier in ocular microbiology. We know that clinical features, direct smear examination, and culture are the pillars of diagnosis of corneal infections, and I belong to this school. But in recent times, we have been moving away, or at least we have been hearing that we need to move away. Why is that? We are realizing the limitations of conventional in vitro culture methods. It's a low sensitivity and it takes time. And we also find that over time, in this publication we have shown that culture negatives are increasing. And new studies like using next generation sequencing, they show us that it's not only one organism or two organisms, there are multiple organisms present in an infection. So what do we do with this information that we are getting? We are moving towards that. The advantages are they're very highly sensitive methods and very, very specific as well. And they give you reliable confirmation of new organisms. They also confirm the presence of organisms which are difficult to culture. So these are the advantages that we may have. And the basic of this is all DNA of all organisms are similar. DNA is same. You can have different methods, and one of that is nucleic acid probes are available to identify. Now these are targeted. You need to know which organism you are looking for, and then you use the probe for that. PCR is also targeted. Usually you amplify the DNA and then identify that in a gel electrophoresis, and you can find the presence of the DNA. There are many modifications of this PCR. Multiplexing will just increase the number of targets. It will not be all, but some of them. And you can have nested PCR to increase the sensitivity. And I'll go through the others in next slides. DNA chip, we are very proud that in India, our first DNA chip for ocular infections was made by Exciton, and we had worked along with the RP center with them. They actually have a center in Bangalore, and you can send samples there. They use the technique of multiplex PCR with hybridization and give you the report within 24 to 48 hours. We all learned about real-time PCR during COVID. Whether you knew or not, everyone was talking about real-time PCR, and we know that it's a very, very sensitive method for diagnosis of viral infections. A lot of work is going on about DNA sequence. We are not stopping at finding DNA is present. We are looking at the parts of the DNA. What are the ATCG sequences in that part of the DNA that we are dealing with? And that's what is called DNA sequencing. Very beautiful methods, very quick methods are available to now find out the sequences, which to make it so simple, you just see 4 colors, A, T, C, G, it tells you, and you get alphabetical, you can see a whole long chain of alphabets of A, T, C, G, tells you what is the sequence of these bases in the DNA sample. And then there are software which will tell you which organism you are dealing with just using that sequence. There were many studies. Actually, 2008 itself, from Aravind, we had studies to show that PCR is more sensitive than culture. And we also showed that real-time PCR is more sensitive than uniplex PCR or targeted PCR. Sequencing helped us really to catch the pythium that we have been talking about. So it is used for identification of different species. And here is in a phylogenetic analysis, you find that we were actually dealing with pythium, what we were not able to identify. And retrospectively, we learned what is the smear looks like, what does the culture looks like, and also we learned the formation of zoospores, which we are extensively using for antibiotic susceptibility testing for the pythium. But we are not satisfied with that. We are now going to next generation sequencing. So what I meant by targeted is a short fragment of DNA, a particular type of DNA, whereas that is overcome in next generation sequencing. You look for any DNA that is present there. You will detect from human DNA to all other DNA that is present, and therefore it is unbiased and untargeted is what it is called, and it is very, very good for identification of unknown organisms in the sample. So this is a massive parallel sequencing that is done to capture large amount of genomic data, and it is less affected by the prior empiric therapy. DNA is not affected. And so that's one advantage. And people are now trying to make bedside setups or tests which can be done in the small laboratories to identify the organisms. There are various types again in this. To a certain extent, you can target just all bacteria or all fungal, and then there is another one which is called shotgun metagenome, which is just to identify any DNA present in the sample. So where are we going with this? There are big machines called Illumina. I was very lucky to just visit Kalyani, which is very close to Calcutta. I never knew there's such a beautiful institute we have, which actually does everything that can be done in genomics. It's a National Institute of Biomedical Genomics, and very, very wonderful. LV Prasad is having some collaborative study with them, and that's why we visited them. So they have everything that is mentioned here, Illumina, pyrosequencing, and but for diagnostically, we are actually more interested in nanopore techniques. They have that too. And not only that, I visited a place in Mumbai IIT where a company called Haystack Analytics, they are actually very close or almost have done for tuberculosis. They do NGS. It can be done in a small laboratory. This is a setup picture I have shown here. It's just a small setup here with a monitor there, and you can actually put the sample, just treat the sample a little bit, put inside that, and you can get the report like this. I have shown here the report shows it's a pathogen, Escherichia coli, and it's got resistant genes. What genes are there in that which is causing the resistance, and you can get a report with all patient information. So they have come up to this stage. It's very, very successful for tuberculosis, and now they're working on urine infection, and we have now tied up with them for ocular infections. So we are moving very close to that. There are also studies and actually published now and in use in the US, a multiplex PCR with the hybridization technique, which is called nanostring, and they have a targeted thing for 48 targets in total, and this is totally ocular panel, which is 29 bacteria, 10 fungi, and it includes viruses as well. So things are really moving fast. Within 12 hours, you can get these results. And a clinical study that has been done in bacterial keratitis is already published. You can have a look at that. Now I come to the limitations of these. We are still learning in this area, technically demanding, and most difficult thing is it is centralized. It's not available to small labs. It's only in big places. You have to send samples and get the results. And a lot of clinical validation is required for these studies, for us to know when we can apply, what information is important for treatment, and what can be just ignored. So their softwares have to be really developed further to make them robust in results.